Welcome to this lesson on Meiosis I, where we explore the process of separating homologous chromosomes. This is the first division in meiosis that plays a crucial role in reducing the chromosome number in cells. Understanding this process is fundamental to grasping how genetic diversity is created in sexually reproducing organisms. Meiosis I is the first division of meiosis where homologous chromosomes separate from each other. This process reduces the chromosome number from diploid, which is 2n, to haploid, which is n because it reduces the chromosome count by half. Meiosis I is known as the reductional division. This is a critical step in producing sex cells or gametes. Meiosis I consists of 4 distinct phases. First is Prophase I, where homologous chromosomes pair up and crossing over occurs. Second is Metaphase I, where the paired chromosomes align at the cell equator. Third is Anaphase I, where homologous chromosomes separate and move to opposite poles. Finally, Telophase I results in the formation of 2 haploid cells. Each phase has specific events that contribute to genetic diversity. Prophase I is the longest and most complex phase. 4 key events occur during this phase. First, chromatin condenses into visible chromosomes. Second, homologous chromosomes pair up in a process called synapsis. Third, crossing over exchanges genetic material between paired chromosomes. Fourth, the nuclear envelope breaks down, allowing spindle fibers to access the chromosomes. These events are essential for genetic recombination. Synapsis is the pairing of homologous chromosomes during Prophase I. When 2 homologous chromosomes pair up, they form a structure called a tetrad, also known as a bivalent. A tetrad consists of 4 chromatids held together. These paired chromosomes are held together by a protein structure called the synaptomal complex. This pairing is essential for the next critical event, crossing over. Crossing over, also called recombination, is the exchange of genetic material between non-sister chromatids of homologous chromosomes. This exchange occurs at specific points called chiasmata, which is the plural of chiasma. Crossing over creates genetic variation in offspring by producing new combinations of alleles. This process is one of the primary mechanisms that ensures each gamete carries a unique set of genetic information. During metaphase I, homologous chromosome pairs align at the metaphase plate, which is the cell's equator. Spindle fibers attach to structures called kinetochores on each homologous chromosome. An important process called independent assortment occurs here, where chromosome pairs orient randomly. This random orientation creates additional genetic variation because maternal and paternal chromosomes can face either pole independently of other pairs. Anaphase I is when homologous chromosomes separate and move to opposite poles of the cell. It is crucial to note that sister chromatids remain attached at their centromeres during this phase. The spindle fibers pull entire chromosomes, not individual chromatids. This is a key difference from mitosis, where sister chromatids separate. In meiosis I, homologs separate, which is what reduces the chromosome number. During telophase I, chromosomes arrive at opposite poles of the cell. Nuclear envelopes may reform around each set of chromosomes. Cytokinesis then divides the cytoplasm, creating 2 separate cells. The result is 2 haploid daughter cells. Each cell contains 1 chromosome from each homologous pair. These cells are genetically unique due to crossing over and independent assortment that occurred in earlier phases. Let's review the key takeaways from meiosis I. First, meiosis I is the reductional division that reduces chromosome number from diploid to haploid. Second, homologous chromosomes separate, not sister chromatids. Third, crossing over and independent assortment create genetic variation. Fourth, the process results in 2 haploid cells with unique genetic combinations. Finally, sister chromatids remain together until meiosis II. Understanding these concepts is essential for comprehending how genetic diversity is generated in sexual reproduction.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.